Researchers studying the gut microbiome during Ramadan fasting in 2019 expected to find what most intuition would predict: remove food from the gut for extended periods and the microbial ecosystem should suffer. Diversity should drop. Beneficial bacteria should decline from lack of substrate. The ecosystem should be stressed. Instead, they found that specific bacteria, the ones most strongly associated with gut barrier integrity and reduced metabolic inflammation, were among the ones that increased, not despite the absence of food, because of it. The gut microbiome during 72 hours of fasting and autophagy does not behave the way anyone who has only heard about feeding the microbiome would expect. It behaves like an ecosystem that has been waiting for exactly this condition to reset itself. Think of the gut microbiome as a forest ecosystem that changes with the seasons. During spring and summer, the continuous feeding state, the fast-growing opportunistic species dominate. They are fueled by the constant stream of dietary substrate arriving from above. They are numerous, diverse, and faster producing. They crowd every available surface. When autumn arrives and the dietary substrate stream slows, when 72 hours of fasting removes the constant food supply that most gut bacteria have been living on, the ecosystem shifts. The fast-growing dietary-dependent species begin declining. They lose their fuel source. Their numbers fall. And the species that have been waiting in the understory, the ones adapted to survive on the forest floor itself rather than on incoming nutrients, begin to proliferate, not because conditions are good for them in the usual sense, because the competition that was outcompeting them has retreated. The winter of the gut does not destroy the forest. It selects for the species that can survive winter, and those species happen to be among the most beneficial the gut microbiome contains. Here is what the research shows across the 72-hour timeline. Number 1, the dietary substrate bacteria begin to shift. The gut microbiome is broadly divided into populations that rely on different fuel sources. Bacteroidetes and firmicutes, the two dominant phyla in most human guts, are largely fueled by the dietary fiber, polysaccharides, and undigested carbohydrates that arrive from food. Remove that substrate and their relative abundance changes. A pilot study published in PMC in 2025, examining the effects of 10 days of complete fasting on gut microbiota in 13 volunteers, found that the fasting significantly affected gut microbiota diversity and composition. Bacteroidetes decreased by approximately 50% and firmicutes by approximately 34% over the fasting period, while protobacteria expanded by approximately sixfold. The interpretation of these changes requires care. Protobacteria expansion during fasting is not uniformly beneficial. This is a complex shift that includes both neutral and potentially pro-inflammatory species, depending on which members of the phylum expand. The research here is genuinely complex and not fully resolved. What is clear is that the gut ecosystem shifts substantially within the first 72 hours as the dietary substrate changes. The forest is changing, which species dominate the canopy. Number 2, Akkermansia muciniphila proliferates. This is the finding that most directly challenges the intuition that removing food harms the gut. Akkermansia muciniphila is a mucus-degrading bacterium. It feeds on the mucus layer that lines the gut wall rather than on dietary substrate. When food disappears, Akkermansia does not lose its fuel source. Its fuel source is the gut's own mucus lining, which continues to be produced regardless of whether food is present. In research on Ramadan fasting reviewed in Science Direct in 2025, Akkermansia muciniphila was shown to increase by 32% at 4 weeks after Ramadan fasting compared to baseline. The direction of this change is consistent across multiple fasting studies. Akkermansia is specifically adapted to thrive in the conditions that extended fasting creates, and the consequences of this are significant. Akkermansia muciniphila is one of the most extensively studied microbiome species for its association with gut barrier integrity. Higher Akkermansia abundance is consistently associated with reduced intestinal permeability, lower metabolic inflammation, improved insulin sensitivity, and reduced adiposity. It is not a species that most people are trying to feed with supplements. It is a species that thrives when you stop feeding the bacteria that were outcompeting it. The understory species is finding its moment. Number 3, Faecalibacterium prausnitzii shows characteristic changes. Faecalibacterium prausnitzii is the primary butyrate-producing bacterium in the human gut. Butyrate, a short-chain fatty acid produced by bacterial fermentation, is the primary fuel source for colonocytes, the cells lining the colon. It is also one of the most potent endogenous anti-inflammatory molecules produced in the gut, exerting effects both locally in the intestinal mucosa and systemically through the bloodstream. The same Science Direct review found that Faecalibacterium prausnitzii increased by 24% following Ramadan fasting. And further downstream, genes associated with short-chain fatty acid production in the Lactobacillus family, the broader bacterial group that includes butyrate producers, were upregulated, and fecal concentrations of butyrate and propionate rebounded to 25% above baseline levels after the fasting period ended. This is a finding with direct structural implications. The colonocytes that depend on butyrate as their primary fuel were being served by a microbiome that had been recalibrated toward producing more of it. The forest floor is becoming more productive for the species that feed the soil itself. Number 4, gut epithelial autophagy runs and the barrier strengthens. This is the change with the most direct implication for what happens outside the gut, because what occurs in the epithelial cells during 72 hours of autophagy affects the entire system in ways that most gut health conversations never cover. The gut epithelium is the single-cell barrier between the interior of the intestine and the bloodstream. These cells, enterocytes and colonocytes, are among the most rapidly renewing cells in the body, turning over completely every 3 to 5 days. They are also among the most metabolically demanding. Under continuous feeding conditions, they are processing, absorbing, and filtering a constant stream of material, generating significant metabolic waste in the process. Research documented in Trends in Microbiology in 2023 confirmed that during fasting, the intestinal mucosa undergoes renewal concomitantly with changes in immune function and a decreased inflammatory state. Autophagy in these epithelial cells clears the accumulated damaged proteins and dysfunctional mitochondria that have built up from the high-demand processing of continuous feeding. The barrier cells become cleaner. Their junction proteins, the tight junctions that determine how permeable the barrier is to molecules from the gut lumen, function more precisely in a cell that has been autophagically maintained than in one carrying a debris load from continuous metabolic activity. Gut permeability, the degree to which molecules can cross the epithelial barrier inappropriately, is central to the inflammatory conditions associated with modern metabolic disease. Autophagy in the gut epithelium is one of the primary mechanisms by which that barrier maintains its integrity, not through supplementing from outside, through the internal maintenance system that only runs at full capacity when food is absent. The forest floor is not just changing species, the soil structure is being renewed from beneath. Number 5, the gut virome shifts and resets. The gut ecosystem contains not just bacteria, but an entire viral community, bacteriophages, which are viruses that infect and kill bacteria. The phage population in the gut is vast and largely invisible to most discussions of gut health, but it plays a critical role in regulating which bacterial species dominate the microbiome at any given time. Research published in NPJ Biofilms and Microbiomes identified 49 phages showing reproducible directional changes during extended fasting. The study found that fasting induced a transient shift in the gut virome toward increased representation of certain phage types. These phage changes are not random. They are part of the selective pressure that helps determine which bacterial species survive the fasting period and which decline. The 72-hour phage shift is part of a broader ecosystem-level reset. The forest is not just changing which trees are dominant. The entire regulatory system that controls the forest composition is recalibrating. Before the protocol, 72 hours of fasting is extended fasting and should not be attempted without medical guidance and prior experience with shorter fasting windows. If you have diabetes, kidney or liver disease, a history of disordered eating, are pregnant or breastfeeding, or take ongoing medication, speak with your doctor before attempting any extended fast. Electrolyte management throughout the fast is critical. This content describes general microbiome biology and does not treat or diagnose any specific condition. The 5 changes during 72 hours of gut autophagy follow a coherent biological logic. The dietary-dependent bacterial populations shift as their substrate disappears. The mucus-feeding bacteria that protect the gut lining proliferate in the relative vacuum. The butyrate producers recalibrate and emerge from the fast, producing more of the molecule that fuels the gut wall. The epithelial cells run autophagy and the barrier tightens. And the viral regulatory layer recalibrates in parallel. The gut ecosystem during 72 hours of autophagy is not starving, it is selecting. If this changed how you think about what removing food does to the gut, subscribe and leave a like. Tell me in the comments, did you expect the microbiome to suffer during extended fasting, or did you suspect the ecosystem would respond this way?